So I'll kind of focus my time here on giving an introduction to PrimeFlow, and then you'll hear a little bit more on the actual experimental data and the two talks after this as well. And so PrimeFlow is really an application of the branched DNA technology specifically to be used in flow cytometry. So it allows you to take a lot of the advantages of flow, but combine that with the ability to ask questions about gene expression as well. And I think Chris really did a nice job of kind of highlighting a lot of the advantages of flow cytometry, kind of the power of that technique, the different options that are out there for that. And flow really does have kind of a very unique power in the ability to answer questions about individual cells. But traditionally, most options for looking at gene expression are kind of bulk assays. So you're getting answers on, populations of cells or things like that. And I think PrimeFlow really fits kind of nicely between those in allowing you to bridge the gap between technologies that kind of hasn't been there in the past to ask those type of questions. And so this is kind of just plotting out some qPCR data so why this might be important as if it were flow data. And so if you ran a typical qPCR experiment, you might get some data kind of like this, where you're looking at a couple different genes and you get relatively high signals for both of those. But you don't necessarily know, are all your cells expressing both of those genes? Is it some cells blazing for both, not expressing the others? It's tough to get that information without incorporating some sort of a sorting step or other things that add some complexity and potential for error there. And so with PrimeFlow, you really can very easily get around that. You can get that single cell information. So if you have multiple populations, you can see that you might have cells that are negative for both of these transcripts, some single positive populations, and then perhaps your population of interest that does truly express both of those and is high for both transcripts there. And so what we would kind of argue is that, today a lot of the gene expression technologies are really focused on that blended average of events and expression across different cell types. Certainly there is a place for these technologies, in the future to do initial screening, kind of narrow down target lists. But at some point, I think it can be very useful, especially if you want to ask mechanistic questions, what cell types are drugs acting and what cell types are certain pathways important, to be able to say, in this particular cell type, this is where I'm seeing a change in gene expression or some effect from, my treatment or drug or something like that. And so the desire to do this and be able to answer these gene expression questions in flow has been around for a long time. It's not like we were the first people who wanted to do this. But I'd say there are kind of two limitations that made it kind of challenging. One is just getting enough signal that you can detect low expressing genes, the large amount of variance in expression levels throughout the whole genome. So that's kind of the first hurdle to get over. And then the second is really adapting the protocols you need to use to be able to do hybridizations and look at expression with the normal staining protocols for flow, so that you don't add in this ability to answer the gene expression questions, but affect your protein staining in ways that make it untenable as well. And so kind of the assay of PrimeFlow, we've got around both of these. We optimize that protocol, so you get a kit that has all the reagents in it you need, is compatible with the existing protein stains, and then using the branched DNA technology, as Mike introduced, allows you to get that amplification to be able to detect very low expression transcripts in the same assay then as well. And so Mike covered this just a little bit, but I'll just do a quick review here of kind of how branched DNA works in terms of PrimeFlow. So initially, you have your target of interest, the RNA molecule that you want to detect here, and then what we will generate for you is a series of target probes that are specific to that RNA. And so these, as Mike mentions, are split for added specificity here. We'll lay down multiple pairs of these across your RNA, and then after that, we'll do a series of amplification reactions to amplify that signal so we can actually detect it. And so we'll next come in with a pre-amplifier molecule that will hybridize to those target probes. That creates binding sites for a number of additional molecules called amplifiers that will also hybridize. And then those create even more binding sites for the label probes, the actual fluorescent molecules that we'll detect on your flow cytometer then. And so what this kind of boils down to is that in one of these trees, you have spots for about 400 labels. And we typically will lay down about 20 of these bDNA pairs across your RNA. So between those two, we can get about 8,000-fold signal amplification from one molecule. In certain circumstances, we can even increase the number of bDNAs that we lay down if you've got a really low expressing gene. So we can even bump that out to, around 16,000-fold or so if we need to for very low expressing genes or difficult transcripts. And so Mike showed this slide as well, but I'll just cover it again here. Especially for something like flow, minimizing, background and improving sensitivity is a very important issue so that we're not seeing non-specific signal. And so with this, using that split target probe approach with both a right and a left oligo, that tree will only assemble so that next pre-amplifier molecule can only bind if both of those part of the split probe are there. And so down here, what we're seeing with the actual raw data here and then graphs over on the right is that really only when you have both the right and left oligos do you see any sort of amplification. If you have left or right alone for our GAPDH example, we see essentially nothing. If we add in probes for DAPB, a bacterial gene, we don't see any signal, and of course no probe, we don't either. So really it's the combination of the amplification with this split approach for specificity that I think gives us a much, much better signal to noise than you would see with kind of earlier technologies that tried to do this then. And so this is actually showing some data from a publication making that same point. In this case, these researchers, probably because a reviewer had told them to do this, were demonstrating that their probes were specific for human beta-2 microglobulin here. So in this case, they had stained their cells with a human-specific CD45 antibody and then a human-specific beta-2 microglobulin PrimeFlow mRNA probe. And so that data is graphed up here, and you can see that really only for that, actually they mixed together both human and mouse CD4s here to show that even for the exact same cell type, target, they can pick out those subtle differences across species for this target here too then. So just in the human cells, so those that were positive for that human-specific CD45 cell, did they really see any appreciable signal for the beta-2 microglobulin mRNA? So kind of not just in our example, but also in the published literature, the specificity of the assay has been pretty well demonstrated then too. That's just showing those two populations then of the human and the mouse there. And so I won't spend too long on this slide because Mike also put it up briefly. But I think just looking at these middle panels, you can really see the stark contrast between the PrimeFlow approach up here with branched DNA, the specificity, and then down here looking at just kind of a traditional fish assay, where we only have a limited number of probes. Each of those is only attached to a single fluorescent molecule. And so really the difference in signal kind of visually, and then over here when it's plotted out with the signal to noise is pretty apparent and really opens up the ability to look at many more low expressing genes than were feasible with a traditional fish approach there. And so for the actual workflow of the assay, the way it generally works is you would kind of work with us to initially design your probes. We do that process for you with the sequences you send us. We'll also work with you and help you design a panel to make sure it's optimally set up for PrimeFlow as well. Then you can go through, when you're starting the assay, you actually do your surface and intracellular staining as you would normally do. The kit has any required buffers for that already included in it. And then kind of the final step of the assay is actually doing these hybridizations for your RNA transcripts. And so right now we have 4 different probe types. So you can look at as many different protein antibodies as you want to, and then up to 4 different RNAs kind of spaced throughout the spectrum there to be able to combine those 2 approaches together here. And in terms of sensitivity, this varies a little bit depending on the target you're looking at, and certain probe types certainly are more sensitive than others. Our type 1 and our type 10, so the type 1 is the Alexa 647 here, are our most sensitive probe types. And in this case here, we're looking at a gene, HMBS, is involved in the heme biosynthetic pathway. And with those most sensitive protypes, we can usually see down to about 10 copies per cell. And then the other two protypes, 4 and 6, here we're looking at RNA Pol2, are just a little bit less sensitive, but usually somewhere in the range of like 30 copies per cell or so is detectable with those protypes as well. So you don't need to have a lot of copies per cell to be able to look at genes in this assay then too. And so I'll shift gears just a little bit here and kind of go from talking about the basics of the assay into some of the actual applications. And so this is far from exhaustive, but just highlight some of the more common things we found that people are using PrimeFlow for. But as you'd expect, really one of the big uses of the assay is looking at just the heterogeneity of gene expression. So, what cell types genes are being expressed in. Be able to look at the kinetics of transcription along with translation is also something that we see a lot of researchers using the assay for. As Chris mentioned, it's a nice workaround. If you don't have an antibody, you don't have to spend the extra money and wait and then potentially find out the antibody doesn't work. If you're looking at a new target, you can order probes and in a week or two have those and be running the assay against really any gene in the genome. And actually viral work has also been a very popular application of the assay, so we see a lot of researchers using it for that. And finally, I won't show any data for this here, but it is also possible to use the assay for microRNAs too. So we have a slightly modified version of the assay, use the normal kit, and then we just have an additional fixation buffer you can include if you want to look at those instead of just normal mRNA. So I'll show you a few examples from the literature here then before Jason and Valentina show some of their data too. This is one example of a PrimeFlow experiment where researchers were both incorporating a time course to look at transcription and translation, and we're looking at a very rare subtype that would be kind of tough to look at this another way by sorting or something like that. And so in this case, they were using a mouse prostate cancer model, and they were interested in a subpopulation of IgA high-expressing B cells. And B cells they had found were infiltrating tumors and inhibiting the function of CD8 T cells then. And so they wanted to characterize kind of the expression and some of the phenotype of these cells and turn to PrimeFlow to be able to do that. And so in this case, initially on the left here, they just did a positive control, staining for CD45 and then beta-actin as their positive control gene, seeing that essentially all of the cells there are expressing that that transcript, so the assay is working well there. And then they wanted to look at IL-10 because they thought this was involved in that inhibitory phenotype of these B cells. And so they stained for IL-10 protein and then also included a PrimeFlow probe for IL-10 mRNA here. And they did a stimulation with PMA, ionomycin, and LPS, either a 1 or 5 hour stimulation. And you can see initially at the 1 hour stimulation, they can see the message coming up first, but not really a lot of protein. And then at the later time point there, they're still seeing some message, but now also the protein is coming up too. So I think this nicely illustrates kind of some of the power of being able to look at message levels in addition to just protein, depending on the time points you're looking at for your assay. And then down here, it kind of highlights too just how rare this population of cells that they were looking at. This kind of goes through the gating that they worked out for all of these, but in a given mouse, they might be looking at as few as 500 cells here. And so going with other approaches to try to sort or things like that could be tricky if you don't have a large amount of input that you're starting at and potentially losing cells at each of these steps to that. So PrimeFlow could be a nice workaround in those situations. Similarly, in another paper here, these researchers also wanted to kind of get around a tricky sorting issue to look at a specific population of cells. So these researchers were looking at samples from patients that had non-small cell lung cancer, and they were interested in characterizing the FOXP3 positive population of cells. So these Treg populations that are involved in kind of blocking some of the immune response to these tumors and inhibiting that. It can be tough to sort these cells out and get a completely pure population for technical reasons, which is kind of limited abilities to characterize and phenotype these cells to some extent. And so they turn to PrimeFlow as a way to not have to worry about doing any sort of a sort step. They just use a PrimeFlow probe for FOXP3 here. You can see that worked well. They were able to separate their FOXP3 positive Tregs from the effector T cell population. That's also graphed out there on the right with the FOXP3 mRNA on the bottom here, and on the y-axis the protein staining there, just demonstrating they were able to pull that population out. But then for the actual kind of interesting part of their study, they wanted to move on and look at some candidate genes to see what other genes were also being upregulated in tumors that they were taking from these patients versus either healthy lung tissue or PBMCs and lymph nodes that might be also involved in kind of this FOXP3 positive, this Treg phenotype. And so they compared ratios of expressions in the tumors versus healthy lung and control cells here. Of course, they can see upregulation of FOXP3 mRNA in these cells, but they also identified 3 other genes that seem to be important as well that also might be contributing to this phenotype. And so PrimeFlow is a way they could really confirm with a lack of ambiguity that these other transcripts were also present in these cells, and then kind of follow up that in future studies too. Going back to the idea of using PrimeFlow as a workaround for not having an antibody available, this is a paper from Nature Communications back in 2014. These researchers were studying idiopathic thrombocytopenic purpura, which is a condition where antibodies attack platelets, and IL-21 is implicated in the pathology of this. And so they wanted to look at levels of IL-21 protein, and they were also here looking at IL-2 as well. There are, plenty of good IL-2 antibodies available, but at the time there were, not many good options for IL-21. And so when they did their simulation here, they were able to see nice IL-2 signal, but really nothing appreciable for IL-21 just because of the, the lack of quality reagents at that time. And so to get their paper published, rather than waiting around to test a ton of different antibodies, they turned to PrimeFlow as a workaround for that. And so here they're looking at, instead of protein, IL-2 mRNA, and then that IL-21 mRNA probe in place of the antibody. And now when they do the stimulation, you can see not only do they see a nice IL-2 signal, but they're also being able to pick up IL-21 positive cells as well here. And so, this is one example of a case when they use this. We've had people do this for, a variety of unique models, bats, catfish, lots of other things where, there just aren't a lot of antibodies available. So this can be kind of a nice quick way if you need to get an answer fast, rather than, waiting to generate your own antibody or something like that. And then kind of one additional example I'll show here, highlighting the viral applications for PrimeFlow. In this case, these researchers were studying a co-infection model between Epstein-Barr virus and Kaposi sarcoma virus. And they were looking to see if co-infection with both of these viruses, they knew it was promoting tumorigenesis, and they wanted to see if co-infection was actually increasing some of these Epstein-Barr associated RNAs that were involved in cell lysis then. And so they had tagged their KFHV virus with GFP so they could pull that out. And then were infecting with various combinations of these virus and looking for expression levels using a PrimeFlow probe for these Epstein-Barr associated lytic transcripts then. So you can see with PBS, we see very low levels of these. KFHV, same thing. When they infect with Epstein-Barr, they see a jump in those transcripts as you would expect, but that's really even more pronounced when you co-infect with both of those viruses. And when they go on to not only co-infect with both, but also gate on just the live GFP positive. So this GFP positive are the cells that are positive for KSHV. They can see even a greater enrichment of cells expressing these transcripts in those populations too, and that's quantified down here. So this is kind of one a little bit more cancer-focused example on how people are using PrimeFlow to look at viral RNA. But we've got lots of researchers studying other viruses. HIV is really an important and pretty popular method that lots of researchers are using PrimeFlow for, just because of that sensitivity. So we've got lots of examples in the literature of that too. And so one final example I'll show here, I've highlighted a lot of different examples of looking at RNA expression, but it is possible also to look at telomere sequences too. And so in this case, we're generating probes against the single-stranded telomere DNA here at the ends of the chromosomes, and we can compare the signal from those telomere-specific probes, which is in blue here, to a no-probe control in red, to get an idea of the relative lengths of the telomeres, either across different donors, or in this case, also in two different cell types, both lymphs and then monocytes as well. So just one other assay that's also possible with PrimeFlow there too. And if we actually image these cells after we've done the PrimeFlow staining, you're looking at DAPI, CD3, and then the actual telomere-specific PrimeFlow probe signal then as well. You can see that that signal overlaps nicely with the nucleus, so we can have confidence, we actually are detecting what we think we are there too. And so that is kind of all the examples I'll go over. Just really quickly to wrap up here, if you're interested in running PrimeFlow, we've got a number of people who have done it at OSCR here. Here's really all the equipment that you need, and all of that is available here too. You just need an incubator to hold that temperature for hybridization, a refrigerated swing bucket centrifuge for some of the wash steps, flow cytometer that can detect the signals and whatever else you want in your panel, and then also a temperature probe. Temperature is really the key thing of the assay, being able to hold that temperature very accurately, and so validating that as we did too when we ran it a little bit earlier is really important for getting nice results then as well. And then if you want to order any of these probes, we actually have a PrimeFlow website, so just thermofisher.com/PrimeFlow. You can search all the probes that we have available through that and pick the different probe types and fluorophore options there. But then if you want to generate something that we don't have a probe for, all you need to do is email us sequence or accession number for your gene of interest, and then what color you want that on, and we can turn that around very quickly. Ankit, the guy who does our bioinformatics, is really, really good. Often we'll email back in 5 or 10 minutes with the probes design, so that the turnaround for that is very fast then as well. Yeah, so that's all I've got here, but yeah, thanks everybody, and I'm happy to chat more if you have any questions about PrimeFlow afterwards too then.